Greetings, we are Group 13 and our topic is rice immunity against rice blast disease and effective strategies to control it. My group members are Iman, Imran, myself Rishi, I Nazurah, Suraya and also Caudron Romane. In this presentation, we're going to have a look on the case analysis of rice blast disease, the biochemical pathways in the rice immunity against rice blast disease, the effective strategies to control it and also the outcomes of applying those strategies. Rice agriculture is vulnerable to various biotic and biotic stresses. Fungal pathogens are notable biotic stresses that possess considerable risk leading to disease outbreaks, reduced crop yields, and higher production expenses. An example of such threat is the rice blast disease caused by the Magnaporta oreaceae. The pathogen is spread through wind, contaminated plant remnants, or seeds that are left in the fields. It impacts all phases of rice growth, induces symptoms in several rice parts, including the leaf, collar, neck, panicle, and even the glumis. Magnaporta oreaceae results in a reduction of crop production by 10 to 30% in rice-growing countries, and it poses a significant risk to the food and nutritional security of the rapidly growing global population.
So now let's delve into rice blast disease in Malaysia. So in Malaysia, the focus is on addressing the challenges with the panicle blast impacting the rice yield, even resistant varieties like MR219. So these issues has caused a substantial losses, prompting a need to explore combating its complexities. To counter panicle blast, Mardi has introduced new varieties like MR253 and MR284, with each designed to combat the disease. Despite effort, recurring outbreak persists, highlighting the ongoing vulnerability of the rice blast in the region. Marty's persistent effort underscore a commitment to finding a sustainable solution. Advocacy for a comprehensive disease management approach is growing, emphasizing the need for a holistic strategy in ensuring sustainable rice cultivation. In this united stance, a commitment emerged to overcome the challenges and secure the future of the rice production in Malaysia. The journey reflects a continuous battle against a panic blast, emphasizing the importance of a collective effort in facing this persistent challenge. Magna Proteoraceae, the trigger player causing the rice blast disease, has a fascinating infection game plan. Its hemibiotic strategies mix in both friendly growth and destructive attack. Instead of a straightforward approach, M. origin smartly shift from being a good guest inside leaving the host cell to cause damage like a fool, adapting its plan based on the host plant's stage. Now, let's get into a nitty-gritty of how MRZ pull off these clever strategies. It's all kicked off with a germ tube differentiation, where a special germ tube form, getting the fungus ready for its host interaction. Moving forward, we hit the critical stage of aprisorium formation. So MRZ craft a powerful melanized aprisorium. Basically, it's an anchor to stick onto a rice leaf. But this structure is not just about holding on, it's about the powerhouse for the next penetration stage. So once attached, the fungus bring out the big gun with penetration pack and hyphae wood. A tiny pack pushed by the fungus pressure pierced the plant's layer. So this not only break the plant's defense but also act as a highway for the infectious growth, letting the mRNA travel through the space inside and between the rice plant to the fascinating modes of spread in the rims of the rice plants. Imagine a tiny spore, conidia, produced within the infected rice selection. This spore can act as a nature airborne carrier carried by the wind to the neighboring crops. It's like the fungus catching a ride on the breeze, showcasing its adaptability and ability to move through the air. Now, let's talk about the waterborne transmission. So, when rainfall or irrigation occur, the water droplet loaded with the conidia can transport the fungal spore. This becomes especially influential in the moist condition, using water as a carrier to rapidly spread the fungus to healthy rice plant. So, it's underlined how the environmental factors like rain and irrigation significantly impact the disease spread. Moving on to the seed transmission, a fascinating yet concerning pathway. So when rice seed carry the fungal pathogen, the disease can persist across the planting season, leaving a legacy in the seed.
Now let's have a look into the biochemical process involved in rice innate immunity against rice blast disease. Animals have adaptive immunity, but plants lack specific cells and a circulatory system. So plant cells defend against those pathogens individually through their innate immunity. There are two layers of innate immunity: palm-triggered immunity (PTI), the first line defense, and effector-triggered immunity (ETI), the second line defense. However, the resistance provided by ETI is often overcome in 3 to 5 years due to the blast fungus rapid evolution and great diversity. Therefore, defensive regulated genes in the defensive mediated immunity provide partial but persistent resistance to M. oreaceae infection. So the first one is the PAM-triggered immunity PTI response, which is the first line defense. PTI activates plants' immunity by recognizing highly conserved pathogen-associated molecular patterns (PAMs) using plasma membrane-bound pattern recognition receptors (PRR). In rice blast disease, chitin acts as a pump as it's an integral building block of the fungal cell wall. Meanwhile, rice transmembrane lysine motif receptor-like proteins, which consist of two lysine motif-containing proteins 4 and 6 and a chitin elicitor binding protein, will act as a pattern recognition receptors. This receptor complex from the cell membrane then senses and transmit the fungal chitin-derived signal. After detecting the chitin, the signal will be transmitted via two kind of pathway. The first one is using receptor-like cytoplasmic kinase. After series of phosphorylation, the signal will be transmitted from rice NKK4 protein to MPK36, which will control the defensive responses. The second pathway is by RAG-GF1. Lastly, the RAG active immunity 1 will be activated, which will increase the rice transcription factor and also phenylalanine ammonia lyase 1. So the phenylalanine ammonia lyase 1 will causes accumulation in lignin, and the rice transcription factor will increase the expression of pathogenesis related PR gene. So this gene will produce the antimicrobial that will destroy the fungal cell walls. Magnaporta Oriaceae have developed a method to inject the effectors to subvert the PTI of the rice. So these kind of effectors are known as virulence proteins. So one of the example of a virulent protein is the host transcriptional reprogramming 1 and 2, so which will be transported into the rice nucleus to reprogram the host immune responsive genes and eliminate the host immunity. To combat this, the rice will use the second layer of immunity. In the ETI, the disease-resistant protein R must detect those pathogens, and these R protein are coming from the nucleotide binding and leucine-rich repeat family. There are three kinds of interaction where the molecular mechanism of ABR recognition by an NLR protein is carried out. So this NLR protein, or also known as resistant protein. So the first one is the direct interaction. The second one will be the integrated decoy model, which in two pairs of NLR proteins, such as the RGA4 and RGA5, will detect the effectors such as the ABR-PR or ABR-Co39. And the last one is the indirect model, where 12 ABR-PST interacting proteins are interacted with the effectors. After detecting the pathogens effectors, the virulence proteins, the rice NLR proteins such as PID3, PIT, and PICM-R will cause certain defensive responses. For instance, PID3 will cause the expression of defense-associated genes, PIT NLR will cause the reactive oxygen species production, and lastly, PICM-R will cause the activation or expression of rice defense genes. So the last one is the defense regulator mediated immunity. This kind of immunity will provide partial but persistent resistance against the Magnaportia oriaceae compared to the ETR. This is because the blast fungus rapid evolution and also the diversity. So in this immunity, the signal will be transducted via the APK cascade, ubiquitination mediated pathway, and lastly modulated by transcription factors. After the signal transduction, there will be downstream immune responses. The first one is the reactive oxygen burst caused by the lesion mimic mutant genes and also the microRNAs. So hydrogen peroxide may trigger programmed plant cell death during the hypersensitive responses that may restrict the spread of infection. And the second one is the antimicrobial products. So during the ethylene burst, there will be cyanide production. So cyanide will inhibit the cyanide-sensitive respiration in fungi.
There are few effective strategies to overcome rust blast disease. First, we can do nutrient management by managing nitrogen, water, and crop rotation. Excessive nitrogen can lead to large vegetative growth, which induces the growth of pathogen. Water managing is crucial to avoid growth of pathogen and promote overall plant health, while alternating rice cultivation with other crops can disrupt the life cycles of pathogens. Next, silicon administration can enhance the plant cell strength by reinforcing the cell wall, providing resistance to rice blast fungus. Trichoderma hygienum can also act as a biological control for the rice blast disease. It competes with the menopause oryzae for nutrient and space in the rhizosphere. It uses microparasitic mechanism by releasing enzymes and metabolites to limit the growth of menopause oryzae. It also boosts the plant immunological response to invaders and activate the defense genes of the plant. Bacillus subtilis, which also one of the biological control of this disease, which they will do antagonistic activity against menopause oryzae. This disrupts the pathogen physiological processes. Bacillus subtilis also forms biofilms that serve as protective shield to prevent the entry of fungal pathogens.
In the fight against rice blight disease, chemical control is still the most effective approach, even if it has been criticized for resistance or environmental concern. Recent research tested combinations of fungicides like azoxystrobin and triazole, and this proved very effective against rice blight disease. So these new combinations could replace tricyclazole and its associated problems. But how do these fungicides work biochemically? Well, they target the fungus in 3 different ways. The first one is the membrane disruption, where triazoles will block the fungus from making ergosterol, which is a key component of its cell walls, weakening them and then killing the fungus. The second one is the energy blockage, where strobilurin and triazole mess with the fungus energy production by targeting the electron transport chain in fungal mitochondria and then crippling its ability to spread. The third one is the enzyme inhibition, because some fungicides target specific enzymes the fungus needs, preventing vital processes and then halting its growth. So while concerns remain about overuse and environmental impact, this research shows promise for controlling blast disease and protecting rice crops.
In my part, I'm going to talk about the outcomes of the effective strategies on controlling rice blight disease. The outcome of nutrient management such as nitrogen and nitrates, the color of the plant will change to normal color for the rice plant, and the stem will become thicker. This is the example of the plant without the nitrogen and the plant with the nitrogen. Is the outcome of the silicon treatment. The layer of silicon impairs the growth of robust stems, roots, and leaves. Additionally, if it interferes with the development of a thick layer of silicate epidermal cells, leaving rice plants vulnerable to bacterial, fungal, and insect problems. Those outcomes, after paying a big attention towards the silicon level, we might reduce chalky grain, reduce grain discoloration, increase number of spiklet, grain weight, and fee grain. It also an advantage element to plant development, has demonstrated a preventive effect against many pathogens. This is the example: the plant with the silicon deficiency and the plant when treated with the silicon. Next, the outcome of the biological control of rice blight disease with Trichoderma harzianum. Firstly, there is the outcome of measuring the Trichoderma spp filamentous fungus known as Trichoderma species inhabits soil and includes species that exhibit antagonistic activity against plant disease, including M. oryzae. The production of serum metabolites with antibiotic activity such as peptides, polypeptides, polypeptides, pyrons, and terpenes is a well-known characteristic of Trichoderma species. The harzianum relies on competitive exclusion as a primary defense mechanism against rice blight disease. Trichoderma suppress M. oryzae in acquiring nutrients and occupying space in the rhizosphere. Next, the outcomes of biological control of rice blight disease with Bacillus subtilis. Additionally, utilization of Bacillus subtilis produces biofilms on plant roots that enhance the antibacterial action of lipopeptides in the soil and aid in the production. It can be seen in the picture where the seed treated with Bacillus subtilis, and when the seed is treated with control, there is a difference with the length of the roots. Lastly, the outcome of using chemical control of rice blight disease. For chemical control, it is suggested that azoxystrobin difenoconazole provide disease control. It enhances crop healthy, quality, and production. It can be seen in the picture that before it be treated with the azoxystrobin difenoconazole, the color, the plant is not healthy. When after it used the azoxystrobin difenoconazole, the plant become healthier, more green, and the quality become more rich.
Hence, in this presentation, we have seen the biochemical process behind the rice immunity. And we also have looked how rice blight disease can be prevented with effective strategies and their outcomes. Over half of the world's population relies on rice as their staple food and primary nutrient source. So, this will ensure higher yield of rice, which will strengthen the global food security if we apply these effective strategies. Thank you.